Thank you everyone for joining us, and welcome to the Cancer Research Institute's webinar series, Breakthroughs in Cancer Immunotherapy. Today's date is Tuesday, June 16th, 2014. The title for today's webinar is Developing Effective Immunotherapies for Ovarian Cancer and Other Tumors. Before we begin, I would like to acknowledge our sponsor. Today's webinar is made possible through the generous support of Agenus. Agenus is an immuno-oncology company developing a portfolio of checkpoint modulators, heat shock protein vaccines, and adjuvants. Agenus's checkpoint modulator programs target GITR, OX40, CTLA4, LAG3, TIM3, and PD1. The company's proprietary discovery engine RetroCyte display is used to generate fully human therapeutic antibody drug candidates. Agenus's heat shock protein vaccines for cancer and infectious diseases are in phase 2 trials, and the company's QS21 Stimulon adjuvant platform is extensively partnered with GlaxoSmithKline and Janssen and includes several candidates in phase 3. We thank you, Agenus, for your sponsorship of today's webinar. My name is Brian Brewer, and I'm Director of Marketing and Communications at the Cancer Research Institute. CRI is the world's only nonprofit organization dedicated exclusively to harnessing the immune system's power to conquer all cancers. We fund scientists all around the world, and their work has led to significant breakthroughs in treating cancer with immunotherapy. Over the next 45 minutes, you'll have an opportunity to hear firsthand from an immunotherapy expert how cancer treatment is undergoing a revolution thanks to these breakthroughs. After a 25-minute presentation from our speaker, we will open up the discussion to questions submitted by you. You can submit your questions at any time by using the Q&A box on your screen. Today's webinar is being recorded and will be made available for viewing on the Cancer Research Institute website. Now it's my pleasure to welcome today's expert speaker. Dr. George Coukos is Director of the Department of Oncology at the CHUV in Lausanne and Director of the Service of Developmental Therapeutics. He's also the Director of the Ludwig Centre at Lausanne in Switzerland. He's a recognized global leader in ovarian cancer immunotherapy and tumor microenvironment research. His research focuses on understanding the immune system's response to ovarian cancer and other tumor types, and on developing therapies that enhance that response. He's also developing tools to target the tumor vasculature. Dr. Coukos also has extensive experience in designing and conducting phase 1 clinical cell-based immunotherapy studies and in the development of cell-based immunotherapy from the laboratory to the clinic. So now it's my pleasure, Dr. Coukos, welcome.
Thank you very much, Brian. It's indeed a pleasure and an honor to be hosted by the Cancer Research Institute series in webinars in cancer immunotherapy. This is indeed a spectacular time for cancer immunotherapy, and it is so also thanks to the effortless, to the relentless effort of the CRI, and of course the long investments by its partner, the Ludwig Cancer Research and the Ludwig Institute. So I'd like to present today the landscape of immunotherapy in ovarian cancer, at least from our point of view, as we have been developing it in the last few years. So ovarian cancer remains still a very important problem. We've with other experts recently had the opportunity to convene and think through the challenges in ovarian cancer, which are very explicitly illustrated in this slide, where you can see that basically survival in 3 international large patient series has not been impacted over the last few decades, in spite of our significant efforts in escalating chemotherapy and identifying new cytotoxic and therapeutic agents. And therefore a lot remains to be done in this domain. Of great disappointment came the recent data of the TCGA that basically, after having analyzed approximately 490 high serous ovarian tumors, revealed that there are no druggable obvious specific targets in ovarian cancer other than the ones we knew, which is basically BRCA1 in a proportion of patients, and the whole homologous repair recombination engine, which is broken down in some of these patients. and of course p53, which is very early and inevitably lost in ovarian cancer, and a few other pathways, but no obvious drivers that can lead to obvious therapeutic interventions in the short term. And so therefore we must find solutions for these patients. We and others have, of course, turned our attention a decade ago to the tumor microenvironment, as we view this as an interesting opportunity for understanding biology and deriving therapeutic targets. And this slide here illustrates the view as it came through the visionary work by Doug Hanahan and Bob Weinberg, illustrating the significance over a decade and a half ago of the tumor microenvironment. In almost 10 years ago, we described the classification of ovarian cancer based on immunophenotype, if you will, derived by the pattern of infiltration of T cells in the tumor microenvironment. Here on the right side, in this picture, you see in immunohistochemistry with CD3, every one of these dots is a T cell infiltrating ovarian cancer, and you can see T cells infiltrating the epithelial component of the tumor islets over here and here, as well as the stroma, the intervening stroma here. So this is one pattern of infiltration. We call these intratumoral T cells or intraepithelial T cells. Contrast this to this pattern where the T cells are present in the stroma, but there are no T cells in the tumor islets. And so these tumors have paucity or no intraepithelial or intratumoral T cells. And so we ask the question of whether this in fact correlated with different outcomes. And in fact, we found that patients who had intratumoral T cells at the time of diagnosis were those who enjoyed a longer-term progression-free survival or overall survival, meaning progression-free survival, meaning that they had a disease-free interval since the completion of chemotherapy until the relapse of their disease. And what is quite over here, you can see that about 50% of the patients who had intratumoral T cell actually never recurred, and these are stage 3, stage 4 ovarian cancer patients. And if you look at this over here, this slide, you'll see that in this population of patients where one expects a long-term survivor population of about 20-25%, all of these long-term survivors cluster within the population of patients who had intratumoral T cells at the time of presentation. And in spite of optimal debulking and complete response to therapy, none of the patients who had no evidence of intratumoral T cells survive past 5 years. And so that to us indicated that there must be some very potent biology underlying this, and perhaps tumor immune recognition was a very important determinant of clinical outcomes, which was confirmed recently by this meta-analysis on about 1,800 patients from about 10 studies, all of which showed the same results, and that is the absence of intratumoral T cells correlates with basically an increased risk of death and shorter survival with a hazard ratio between 2 and 2.5. So these cellular biomarkers were recently confirmed also with molecular biomarkers by gene signature that was identified initially by David Botel, and further confirmed by this meta-analysis of an Australian cohort as well as the TCGA cohort, illustrating that patients, in fact, who had this immune signature in their tumors were the ones that performed the best in this green line here. So this biology is quite robust, and it makes us think that, in fact, the presence of an immune response slows down tumor progression and leads to improved outcomes. And therefore, it goes without saying that perhaps the first hypothesis at the clinical level is that if we could activate anti-tumor immune response, maybe we could capitalize and lead to therapeutic gains. So we turn our attention to whole tumor vaccines a few years ago with the notion that whole tumor vaccines encompass all of the possible tumor antigens that are expressed by a specific tumor, bypass limitations that were previously seen by molecularly defined single antigen vaccines, and patients are given the opportunity to be vaccinated against their own tumor-associated antigens. knowing today that, of course, with the knowledge that most solid tumors, and in fact including ovarian cancer, harbor a variety of non-synonymous mutations that are, for the most part, entirely private. And so vaccinating with autologous tumor is what would make the most sense, as an autologous vaccine would encompass all of these possible immunogenic mutated epitopes. And in fact, a meta-analysis performed by Neller over 3,500 patients with putting together data from 173 trials demonstrated that whole tumor antigen vaccines gave the best type of response that one could hope out of this kind of vaccines. So we devised a vaccine a few years ago at the University of Pennsylvania, where we basically harvested autologous tumor from patients with advanced ovarian cancer. These were patients with relapsed, for the most part, platinum-resistant disease. But as long as these patients were eligible to tumor debulking and a good performance status, we subject them to this invasive procedure, of course, harvesting tumor, which was then stored away. Patients were given the opportunity to receive some chemotherapy. And then when they were ready for vaccine, we pulled the tumor out of the freezer, processed it through hypochlorous acid to derive a more immunogenic lysate, which was then pulsed on their autologous dendritic cells, which were then matured with LPS interferon gamma and injected intranodally into groin lymph nodes every 2 to 3 weeks. And that was basically the course of vaccination. We designed a clinical trial in which the patients received in the first cohort only vaccine. And then in a second cohort, we combined that with bevacizumab, with the standard of care dose. The reason why we did the bevacizumab is because of an emerging research line in my laboratory showing that bevacizumab and VEGF blockade basically normalized tumor vasculature and enhanced infiltration of T cells in combination with vaccines into the tumor. And in a third cohort, we combine that also with low-dose cyclophosphamide. This derives primarily out of the work of Liz Jaffe, showing that intermittent low-dose cyclophosphamide has a Treg depleting effect and is quite beneficial in immune interventions. And lastly, a last cohort receives to date actually these two drugs in combination with full-dose aspirin to block prostaglandins, which our research is showing in the tumor microenvironment to be quite deleterious to tumor-infiltrating T cells. And so here's one example from one of the first patients we treated with vaccine alone. This is a patient with stage 2C ovarian cancer who underwent debulking in October 2007. She was then referred to us after chemotherapy because 14 months after surgery and chemo she recurred with a large pelvic tumor, which was resected by us and stored away in the way I described earlier. Received pelvic radiation and chemotherapy, and then recurred again in the omentum here. We resected that tumor as well, and then that was the only tumor that was at the time visible in her abdomen. So we put her on a vaccination process that lasted for almost a year and a half. And then, being this an autologous vaccine, of course, is a finite quantity. The vaccine ended, and she refused to go on any additional therapy because she thought that she was doing great and she was cured. And in fact, she may have been right, because that was 3 years ago, and since then she has been around with no additional therapy and basically disease-free. Nobody knows if she's been cured, but at least at the clinical level, we find no evidence of disease, and that is quite remarkable for advanced ovarian cancer. And these are the earlier results as they're coming out. This is a clinical trial that is conducted at Penn under the leadership of Lana Kandalaft and Janusz Tani, two collaborators who are really driving this vaccine program there. And you can see in green line, this is a control single same hospital population of patients who had advanced ovarian cancer. received at some point during their treatment secondary debulking surgery, followed by bevacizumab and cyclophosphamide at some point. And you see a progression-free survival which beyond 500 days is less than 5%. Compare that with the most recent cohort of patients who are receiving this therapy in addition to vaccine. So secondary debulking surgery plus vaccine and bevacizumab plus cyclophosphamide, with this difference being quite significant. The blue cohort the blue line represents our two earlier cohorts of patients who received vaccine alone or vaccine plus bevacizumab. So the cyclophosphamide, we think, adds more benefit and clearly becomes significant over the control population. Patients who continue to have tumor at the time of completion of the vaccine are then offered the opportunity to harvest peripheral blood, which is stored, and this blood can be utilized for harvesting T cells, which are then expanded and co-stimulated using CD3, CD28. This is a clinical trial that we designed in collaboration with Carl June at Penn. And these cells, after expansion for 11 days in culture in the presence of CD3 and CD28, are reinfused to the patient in an autologous fashion following preparation of the host with high-dose lymphodepleting, non-myeloablating chemotherapy with cyclophosphamide and fludarabine. And here is an example of one of these patients. So this patient had primary debulking in July 2007. She was in a randomized clinical trial in which she ended up receiving standard of care in the control arm, recurred within 26 months. She had a secondary debulking from which we maintained the tumor right here, and then was placed on chemotherapy per standard of care, followed by which we put her on maintenance cyclophosphamide and bevacizumab. And about a year after initiation of chemotherapy, we put her on vaccine alone. And so you can see that she received several months of vaccination. Her CT remained entirely negative. She felt in perfect shape, but C125 continued to go up, and she got very nervous. So we added bevacizumab to her regimen. And she had a delayed response to bevacizumab, implying potentially an immune intervention with a decline in CA125. Her CT continued to remain negative. At this point here, we had a progression by CA125, at which point we infused autologous T cells, co-stimulated, and you can see a progressive decline of CA125. At some point over here, she had a repair of incisional hernia, and at that point the exploring surgeons found very little evidence of tumor, which was really encouraging given this very long course and this behavior of C125. And in the bottom here you see progressively increasing frequency of tumor-reactive T cells in peripheral blood as measured by ELISpot assay against autologous dendritic cells with tumor antigen. So these are some of the preliminary data on this clinical trial. We see that these T cell infusions, most of the patients received one T cell infusion. This is an entirely outpatient regimen. There were no severe adverse events attributable to the T cell transfer. There were, of course, the expected neutropenias and lymphopenias from the high-dose chemotherapy, but overall tolerated extremely well. And we had about 77% clinical benefit in this population, mostly as stable disease. And so we're now working in the laboratory to improve on this approach. And here are the cumulative results on now overall survival. And you can see that this population is now really doing very well. This is the control population of patients in green who received secondary debulking bevacizumab cyclophosphamide. These patients here have received vaccine plus bevacizumab or vaccine alone, and potential adoptive T cell therapy. And these patients here have received vaccine with cyclophosphamide followed by adoptive therapy. And you can see that their overall survival remains stable quite a long time. And so clearly this is now, we think, an interesting and promising approach, and we continue to build on this with further technology. But this cellular-based vaccination, we recognize, are a cumbersome approach, and particularly may not be feasible for every patient because not every patient is, in fact, a surgical patient. And so there may be other ways that one can intervene at the tumor microenvironment to induce the so-called in situ vaccination, so to trigger the same mechanisms of successful antigen presentation in the lymph node, consequently at interventions at the tumor site. And so modelizing this, basically what one needs to achieve this is a death with antigen release at the tumor site with what's called immunogenic cell death, so release of immunogenic antigen, and that can be today achieved by the employment of immunogenic chemotherapies, and in addition, simultaneous activation of antigen-presenting cells. And today we have toll-like receptor agonists and CD40 agonists with which one can achieve this. The resulting dendritic cells picking up immunogenic antigen will hopefully find their way to the lymph node, reproducing in part what we achieve therapeutically by inserting effective dendritic cells into the lymph node, and this could trigger effective T cells that could make their way back into the tumor. So with that in mind, we chose a combination of pegylated liposomal adriamycin, which is a chemotherapy that we are quite lucky to have in the ovarian cancer armamentarium, known to induce immunogenic cell death, and a TLR8 agonist, which is a new compound activating very powerfully myeloid cells. Why adriamycin? This comes from the work of Laurent Vogel and Guido Kromer, demonstrating in fact that anthracyclines induce an immunogenic cell death. This is by virtue of exposing calreticulin on the cell surface, which provides an eat me signal for dendritic cells and maturation signals once the cells are phagocytosed, and that induces activation of dendritic cells and proper antigen presentation. We combine that with a molecule that very sharply activates monocytes as well as myeloid-derived myeloid dendritic cells, you can see here, with very potent upregulation of IL-12 and TNF-alpha very sharply. There is no activation of plasmacytoid dendritic cells. Contrast this with the effects of known TLR7 and TLR9 agonists. And so we use the combination of these pharmacologic two agents to induce immunogenic cell death and in situ vaccination. We tested that in a humanized mouse model of ovarian cancer, in which you can see that the combination of this VTX compound, the TLR8 agonist with Doxil, induced very powerful synergy in this mouse reconstituted with human HLA-2 bone marrow and transplanted with an HLA-2 of ovarian cancer cell line. These were experiments performed by Andrea Facciabene in the lab at Penn, and led to the initiation of a clinical trial in collaboration with the gynecologic oncology group. A phase 1 study was completed and gave some very interesting results in terms of tolerability, but also to interesting complete responses and disease stabilization of 75%. And that was now followed by a phase 2 study, which has just completed enrollment by the GOG, and it's run by Brad Nelson, who is the principal investigator both of the phase 1 and the phase 2, and the study has met accrual in record time, and we're anxiously awaiting the results. Building on this successful translation to the clinic, we pushed further this combination with now adding one additional stimulus into this equation by activating effector cells that might be triggered by in situ vaccination of the TLR agonist and the adriamycin. And now we put into the mix also PD-L1 blockade that seems to us to be the natural partner in this combinatorial immunochemotherapy. And so we were quite lucky that actually the Cancer Research Institute and the Cancer Vaccine Cooperative group endorsed a recent application to perform a phase 1, phase 2 study where we combine now the doxil or pegylated liposomal doxorubicin with the TLR8 agonist and the PD-L1 antibody provided by MedImmune. This is a wonderful collaboration between the Ludwig Institute and the Cancer Research Institute, and we're certainly quite grateful for the generous support that this provides to test this entirely novel idea of immunochemotherapy. So switching gears now beyond vaccination, what can one do? Clearly, in the domain of molecularly defined, so what we presented so far basically mobilizes immunity against a variety of personalized antigens, the majority of which remain unknown for the moment. But in ovarian cancer, there have been a few antigens that have been identified, and vaccines were derived, with the drawback, of course, that a single antigen vaccine can induce escape clones. This is a wonderful experience published by Kunle Dunsi from a Ludwig clinical trial in which patients were vaccinated with NY-ESO-1 peptide, and this was proven to be an immunogenic epitope and a tumor rejection antigen. In fact, several patients had either clinical benefit or manifested response. But you can see here in a progression, upon progression, patients maintain a T-cell infiltration in the tumor but lost expression of the antigen. So a clear evidence of tumor escape. But in this domain of molecularly defined immune targeting of tumors come now to our aid some very powerful tools of basically immune engineering with either T-cell receptors, point in case of the NY-ESO-1. Now we have very important TCRs targeting an HLA-2 epitope, one of the same that was targeted by the vaccine, as well as chimeric antigen receptors. And I'd like to report here the very promising work by Dan Powell at the Ovarian Cancer Research Center at Penn, who demonstrated, in fact, that one can use the folate receptor as a universal target in ovarian cancer by constructing a very powerful CAR engineered T-cell that induces massive regression of tumors that express the folate receptor in a mouse model. And this is now ready for clinical translation, which we are preparing to do at the University of Pennsylvania. So this is, of course, in the domain of being so lucky today as to having multiple approaches to perform immunomodulation in addition to whatever cellular tools we unleash against the tumor. And this is a review of all the possible targets in terms of immune receptors on T cells. And PD-L1, PD-1 is a very important pathway as it's emerging for a variety of tumors, including ovarian cancer. And so this is demonstrated early on by the impact of PD-L1 on outcomes of ovarian cancer. This is a Japanese demonstrating that PD-L1 expressing tumors are associated with worse survival, and corroborated by recent mouse studies demonstrating that, in fact, a blockade of PD-1 or PD-L1, or in combination with CTLA-4 and vaccine, can bring, in fact, significant additive benefit in a mouse model of ovarian cancer, which is quite aggressive. And you can see here that what is quite critical in these interventions is that immune modulation is carried out during early stages of tumor progression, while the tumor continues to have spontaneous tumor infiltrating T cells. These vanish at a later stage of tumor progression, and at that point immune modulation targeting T cells remains ineffective. That's kind of intuitive, yet a concept that has not made it yet to the clinic, with the ability to really measure T-cell infiltration in tumors prior to enrolling patients to PD-L1 blockade or other immunomodulatory blockade. We asked ourselves why, yet another opportunity for immunomodulation, why in some tumors, in ovarian cancer, T cells are perhaps not able to infiltrate in the tumor microenvironment. And our recent work has demonstrated that in fact there are several pathways that are linked to the tumor vasculature that might prevent T cells that might be elicited therapeutically either through vaccines or other maneuvers to home properly and engraft and reject ovarian cancer. And this endothelial barrier is in part mediated by adhesive prohibitive interactions, which are in part mediated again by the endothelin B receptor, or by death ligands that might be upregulated by the tumor vasculature, such as Fas ligand, as well as other co-inhibitory receptors. And very recently, we completed this story demonstrating, in fact, that the tumor vasculature expresses Fas ligand in the tumor microenvironment, and that correlates with the absence of T cells in the tumor microenvironment. But luckily, this is a pathway that one can reverse pharmacologically by blockade of VEGF and prostaglandins, combining anti-VEGF therapy and aspirin, because we found that prostaglandins and VEGF in an additive way maintain expression of Fas ligand in the tumor vasculature. So blocking prostaglandin C2 and VEGF leads to very important pharmacologic effects, which are associated with a massive infiltration of T cells in the tumor microenvironment in the absence of any other immune manipulation. And these experiments really told us that now there are opportunities to recuperate T cell infiltration in those tumors in which none exists at the time of diagnosis. And we have now very interesting therapeutic opportunities to really unleash immunomodulatory therapy together with therapy that counters the tumor endothelial barrier. So as we move forward in ovarian cancer, but these are actually biologies that hold true for a variety of solid tumors, we think that one can articulate a combinatorial approach to immunotherapy. The main goal has been in the last 2 decades to develop methodologies to expand tumor-reactive T cells. This was the case of vaccines for the last 2 decades. This is the case of engineered T cells, which have clearly demonstrated significant potency, particularly in liquid tumors. But one now has a unique opportunity to combine that with disruption of homeostatic regulatory mechanisms, meaning those pathways that are co-opted by tumors to attenuate immune rejection, PD-1, PD-L1 being one of them, but multiple overlapping anergic mechanisms are actually operating the tumor microenvironment, and this should be taken out pharmacologically. And of course, in tumors where we don't have a T cell infiltration at the onset of therapy, perhaps there is an opportunity now to drive in these T cells through angiogenesis blockade. And with that, I would like to really acknowledge the institutes that have supported my work, the wonderful collaborators that have contributed to this important work, as well as the founding agencies. And I will take your questions.
Great. Well, thank you very much for that, Dr. Coukos. Very, very enlightening presentation on immunotherapy for ovarian cancer and other tumors. So we did get some questions from the viewers, and I'll just kind of start into these, and hopefully we can get to as many of them as possible. I will continue to take questions. Also, you can you can type your questions into the Q&A box on your screen, and we'll try to get to as many questions as we can. So the first question is, with so much excitement currently being generated in the space around checkpoint blockade, some of which you discussed in your presentation, what role will vaccines then play? Are they overshadowing vaccines, or is there some other way that these will work together?
Yeah, no, this is a great question and very timely, and in fact that illustrates the painful path of developing vaccines, which have produced reproducibly failures in the past 20 years, but now we understand that many of these failures were in fact due to our inability to energize T cells adequately. Now that we can actually do that, I think vaccines are becoming back and getting under the spotlight, because vaccines uniquely have the ability to expand population of younger cells, which could be more responsive to checkpoint blockade, or at least checkpoint blockade monotherapy. And I think experimental data from multiple murine models in many solid tumors are telling the same story, and that is that there is a significant potential for vaccines to synergize with checkpoint blockade. And I think these experiments are also forthcoming in the human.
Thank you for that. Let's see, are type 1 ovarian cancers, so Ig serous, endometrioid, or clear cell ovarian cancers, more responsive or less responsive to immunotherapy, and if so, why?
Yeah, we don't know the answer to that. So this is something that we need to address as we go forward. As you know, the opportunity to perform this kind of work is primarily with high-grade serous ovarian cancer, because these are the patients who are more frequently found in clinics with relapsed tumors. These are also the patients where we have the opportunity to harvest a large amount of tumor at the beginning, where we perform a lot of these studies. So the answer is, it was a long answer to say we really don't know, and we will have to test that in the future.
Okay, thank you for that. You brought up single antigen vaccines and tumor escape. So if single antigen vaccines lead to tumor escape, does that invalidate the entire approach of single antigen vaccines?
Well, I think that universally single antigen vaccines have shown to be less powerful weapons against tumors than multiple antigen vaccines. So I think that that would be my preferred way of approaching mobilizing immunity against tumors, by basically engaging multiple epitopes at the same time. Now, it's very, very, very interesting to know that we recently had the opportunity to conduct detailed analysis of T cell clonality in anti-tumor T cells recognizing a single epitope. There were hundreds, if not thousands, of TCR sequences that are recognizing one single epitope. So even if one uses one single epitope to vaccinate patients, it doesn't mean that one relies on a single clone of T cells. And I think that, again, further work by Kunle Dunsi has demonstrated that one can elicit different types of clones, and patients with pre-existing immunity have higher avidity clones. A long answer again to say that even a single epitope can elicit multiple T cell clones, but I think in this more is obviously better, and I think multi-antigen vaccination, particularly with personalized, private, and mutated epitopes, if we can find them, or non-self antigens, would be the best approach to vaccination.
Okay, thanks for that. Will all immunotherapy be personalized, or will there be standard off-the-shelf immunotherapy, or is it looking like there'll be a combination of these approaches?
I would think probably a combination. So even so it's clearly desirable to identify a bunch of epitopes, a bunch of antigens that are universally represented in ovarian cancer, and which could be good tumor rejection antigens. The jury is still out. I mean, NY-ESO-1 is clearly an antigen that drives tumor suppression. There's no doubt about that in many patients, but it's not enough, and so one has to now enrich that with additional epitopes. This may lie in the tumor cell, but may also lie in the tumor vasculature in the stroma. And those latter are much more interesting because they are potentially universal tumor antigens.
Okay. Is there any way to find out if a person's tumor has tumor-infiltrating lymphocytes, and depending on that, would the patient expect to either be eligible for a vaccine or immune checkpoint therapy accordingly?
Yeah, so of course there is a way. One can always find archival tumor from the original surgery, or obtain a new biopsy and then stain very rapidly for the T cells. That's very straightforward. And whether this makes patient eligible for one or the other therapy, that remains entirely in the experimental domain. In our clinical vaccine study at Penn, we have not distinguished between patients who have pre-existing or do not have pre-existing immunity. We put everyone in with the expectation that if you can immobilize immunity and you can block the vasculature and other parameters of immunosuppression in the tumor microenvironment, perhaps you can drive a reasonable immune response in any patient. And that's the hypothesis we're going to hope to try to test. So far, we've seen benefit in about 70% of patients, and that exceeds what most studies have shown to be the proportion of patients with pre-existing immunity, which means that perhaps we are changing the microenvironment also in patients who did not have a pre-existing immunity.
Okay, we are almost out of time, but we do have time for another question. What personally, and we get these sorts of questions often from patients who hear about using the immune system to fight cancer, and sometimes there's a misconception that that means if they take high-dose vitamin C, or they eat well, or they rest, or they do yoga, or whatever it is that contributes to healthful living, that that will be enough to boost their immune response and give them protection against cancer. And of course, there are many, many instances of people who live extremely healthy lifestyles and still develop cancer. So I guess this question may be something like that, but let's see. I'll go ahead and put it to you and see how you respond. What personally can a patient with advanced state, high-grade serous ovarian cancer do to jumpstart her immune system?
So that's the holy grail of, whether we can use, commonly available pharmacological or non-pharmacological agents to affect immune homeostasis in the body. I think that still remains an open question. Clearly, there are, I think that the best approach is to try to enroll in a very good clinical trial. So that's a way where things can be monitored, are using scientific rigor to be developed. They have a very strong rationale, and they have undergone very strong peer review by the NIH, by the FDA, and most importantly by the colleagues, ethics committees where patients, patient advocates, and other constituents participate. So I think this has gone the full proof of approval, and my recommendation is to activate the immune system, one must identify a good clinical trial.
Okay, thanks. And for our last question of the day, I'd like to thank everyone for submitting the questions. It's wonderful to see all of you so engaged in Dr. Coukos' presentation. So the last question is, where does ovarian cancer fall on the spectrum of immunogenicity, with melanoma at one end, which has a lot of mutations, mostly due to radiation from the sun, versus leukemia, which may not have as many antigenic mutations? And what does that mean? Go ahead.
I'd like to distinguish between immunogenicity and antigenicity, if you will, or mutational landscape. So if we're talking about mutational landscape, then I think the answer is very clear, because the recent data from Bert Fogelstein and others, there were two very high profile papers that put ovarian cancer right in the middle of all these, of the garden variety solid tumors with an average of about 60 to 70 private and non-synonymous mutations. So contrast that with 1,000 or 3,000 or more that one can find in lung cancer, melanoma, or microsatellite instability, colorectal cancer, or uterine cancer. And contrast that with the very few below 10 that can find in some rare, in some other solid tumors. So we are in the middle, but is that the only source of immunogenic epitopes on the tumor cell surface? Actually, I think that the jury is entirely out, and although the mutational epitopes are, the very fashionable these days, and in a very exciting area, I should add, these may not be the entire truth. And I think that we, by restricting ourselves and only looking for those, we may be missing many of the relevant epitopes that drive T cell recognition and rejection, on which we can build a very strong immune response.
All right. Well, thanks so much for taking the questions, Dr. Coukos. That will conclude today's webinar. Before we leave, though, I'd like to thank once again Agenus for their very generous support of today's webinar. We have one more webinar coming up as part of our Cancer Immunotherapy Month celebrations. We will have Dr. Glenn Dranoff from Dana-Farber Cancer Institute speaking on immunotherapy for lung cancer, which of course is the #1 cancer killer in the United States. As a reminder, I just brought up June is Cancer Immunotherapy Month. We do still have a few more. We're midway through the month now, and we've had a lot of exciting activities which you can check out online to see what we've done so far. And we have a few more good things planned for the near future over the next week or so. So please go to cancerresearch.org/join-the-movement to learn more about Cancer Immunotherapy Month. Also, if you are a cancer patient or a caregiver, or if you are a healthcare professional who talks to patients and caregivers about immunotherapy, or if you are interested in learning more about immunotherapy, we've recently launched a new website called theanswertocancer.org, which features stories told firsthand by patients who've been treated successfully with a broad variety of cancer immunotherapies in a number of cancer types or indications. We also have some insights from oncology nurses and physicians about immunotherapy and how it's transforming their clinical practice today. So again, that's theanswertocancer.org. You can also learn about clinical trials and all the various immunotherapies that are available today, either FDA approved or in clinical trial. Once again, thank you all for joining us. Dr. Coukos especially, thanks to you. And we hope to see you all at next.